Good afternoon, everybody. Thank you for joining Lantern Pharma's Webinar Wednesday. Today, we're going to be talking about childhood cancer. September marks Childhood Cancer Awareness Month. Actually, just the history of Childhood Cancer Awareness Month actually goes back to 1990, when then President Bush actually signed a White House declaration talking about a month set aside to drive awareness in pediatric and cancers. In 2019 actually was the first real time that we actually had a National Childhood Cancer Awareness Month. We thought it was very important to have this webinar at this time because, as many of you may know, we recently were awarded 3 additional rare pediatric disease designations and some of very ultra-rare cancers. Today, I'll be joined with Dr. Reggie, my colleague. He's the head of our clinical development. Reggie and I work closely together on our drug programs and bringing those programs into the clinic. Reggie will be talking a little bit about the challenges faced in each of these tumor systems, providing us details about the patients and about characteristics of these 3 very unusual and ultra-rare pediatric cancers. Before Reggie goes into that, I'll be giving an overview of the stats behind childhood cancer, and I'll also be closing the webinar talking about some of our exciting preclinical and scientific data that is the underpinnings of our 3 disease designations. In terms of childhood cancer, there are about 15,000 new cases in the US that occur in patients that are under 21. And the survival rate overall is about 70%. But it can vary quite drastically. There are many tumors where the survival rates are in the single digit. And there are some that can be easily resolved and managed, such as many of the ALLs and other leukemias. But even given the fact that we've spent a big amount of time understanding and improving the diagnosis, there are only 6 drugs that have been developed for childhood cancer. It's a very small amount. And so anytime you can find new indications, you can find significant activity in these typically rare to ultra-rare cancers, it's actually quite exciting. So for our drug, LP-184, to be able to find now 4 indications is for us very exciting and underscores the importance at getting these drugs into pediatric trials. In the announcement that we made, we talked about 3 rare pediatric disease designations, and any of those designations upon market approval generates a priority review voucher, which then we can sell. And that also is very exciting for not only investors and stockholders, but that priority review voucher sale allows us to then continue developing new programs for additional new cancers. The 3 programs that we were awarded rare pediatric disease designation in. First, malignant rhabdoid tumors, we'll call them MRT at times today. And that was a direct result of the work that we had done earlier in ATRT. If some of you remember, that was our very first rare pediatric disease designation. ATRT and now also MRT, they have a very unique genomic profile marked by SMARCB1 mutation. And that's what really led us from ATRT to the other rhabdoid tumors. We then also found really good and durable response in rhabdomyosarcomas, another ultra-rare tumor, and then also in hepatoblastoma. And so all 3 of these designations came to us at one time. So that's typically very unusual, but the reason we were able to do that, and I'll talk about it a little bit later today's webinar, was driven also by our ability to understand the characteristics of our drug and use AI and in silico modeling to enhance our understanding of where this molecule will work and potentially not work. And then, of course, we were able to get some really very unique PDX models and test out our hypotheses. So we're very excited to advance these 3 programs. Now, my colleague, Dr. Reggie, will start today's detailed discussion, first in malignant rhabdoid tumors. Reggie, do you want to go ahead and give us an overview and talk a little bit about the characteristics of this disease?
Sure, Panna, thanks for that introduction, and welcome you all to this webinar. I thought what I would do is to put in context these diseases. And as much as possible during my presentation, I will talk about the challenges of the current treatment strategies, where there are opportunities for LP-184 based obviously on some of our findings in the preclinical space, as well as what we're also understanding about the drug. So that's the context within which I'll give an overview. So the first indication, malignant rhabdoid tumor. Now, these are tumors that are rare, obviously. That's why we have the designation we're talking about today. But more importantly, they are aggressive. So these are not benign tumors, as it were. Unfortunately, by the time a patient is diagnosed with this disease, it's usually a little bit in the advanced stage. Not surprisingly, these are usually infants. So put it in context, less than 1 year of age and probably about 18 months of age. So it's in early childhood. Now, there are 2 buckets of the disease, those for which we present in the brain or the CNS, which we'll call ATRT. We've just talked about that briefly. And then the second, obviously, outside the brain, the kidneys and soft tissues. Now, the interesting thing is this can spread very quickly, which has its own challenges in terms of treatment. Could we go on to the next slide? Now, these are really ultra rare in terms of the different categories. Upward of about 25 new cases are usually diagnosed in the US. As you can almost imagine, that presents a set of challenges, even for just treatment, but drug development as well. But the age of onset is about 11 to 18 months. That's the median range. Now, what I want you to take away from this is really the 5-year overall survival rate for these patients. About 1/3 of those patients will be alive to see first grade after they are diagnosed. So 2/3 of those patients really will not live to see that. So that gives you the context within which both the regulators and a company like Lantern is really approaching this disease. So go to the next slide. Now, what's the treatment? Current treatment is this bucket of surgery. Obviously, the earlier you see the patient, you can take off the tumor, everybody's happy. And then there's radiation therapy and chemotherapy. These are the main modalities of treatment. Now, the drugs used, there are 3 core drugs: carboplatin, ifosfamide, and etoposide of VP16. Now, let me just remind you, these 3 drugs were not deliberately studied in this indication, but as was the case until recently, most of these drugs are used off-label. There are hardly any really large clinical trials based on the small numbers I've talked about. This is where it gets very exciting for us in Lantern Pharma, because now the question is, if these are the drugs and the standard of care that were not really personalized, yes, they have some benefit, but as I reminded you, more than 2/3 of these patients will not get to see first grade. What can we do better? This is where we come in. While the exact cause is not known, we know there's some genomic underpinning, and that's where really LP-184 might begin to have a role in this disease. We also know that this disease is not as sensitive to this chemotherapy and radiotherapy. Little progress have been made. As I talked about, because of the small numbers, it's very difficult to conduct really large trials. But nevertheless, as we've approached this disease from our preclinical set of data, we're very excited because we see a lot of activity as we use this drug to interrogate some patient-derived sarcoma models. So this is a major reason why Lantern is currently planning with the Children's Oncology Group to undertake early phase studies in patients with ATRT, which again has the same genomic underpinning, and potentially other patients with this SMARCB G1 mutation. The goal here is just really to embrace personalized medicine approach in this space based on what we know currently. We'll go on to the next. This is the ATRT, the atypical teratoid rhabdoid tumors. This one for which is predominant in the CNS. Again, about 58 newly diagnosed patients in the US under 3 years of age. The stats is pretty much the same at about 5 years. These are not encouraging numbers. So our hope is that as we approach this challenge from a personalized medicine approach, that obviously we'll be able to improve in those numbers. So let's switch gear and go to rhabdomyosarcoma. Rhabdomyosarcoma, on the other hand, is a disease that has a bimodal spread. So there's some that occur pretty early, and obviously there's some that occur in teenage years as well. It's a soft tissue sarcoma, and is one of the commonest, if not the most common, soft tissue sarcoma in children and adolescents. Again, the number of cases here, upward of about 500 new cases are diagnosed in the US. Thus, again, the designation that we just obtained. The interesting thing about rhabdomyosarcoma, however, is that histologically, there are different types of this disease. There's the embryonal rhabdomyosarcoma, which accounts for the majority of these patients. And then the alveolar type, about 20% or so. And then you have the undifferentiated, which is also about 20%, and then others in this category. Now, those have some prognostic significance as well as treatment challenges and significance, for the disease. We go on to the next slide. I've talked about is the number and the most common, obviously, in children. But again, I take you to the bottom of the slide. For patients that present early, surgery and treatment, very beneficial. However, a good number of these patients also present with metastatic disease. So either with metastatic disease or when they relapse, it's usually very problematic. Bear in mind the small numbers and the challenge on clinical trials, and of course, some of these agents that are being used for the treatment, which I will talk about later on, as part of the challenges that are faced in this disease and across, obviously, the pediatric oncology space. So how do we treat these patients? Traditionally, again, is the same thing, surgery, radiotherapy, where you can, and then chemotherapy, platinum agents, cisplatin, dactinomycin, daunorubicin, as well as VP16. You begin to see a theme here. These drugs, VP16, they're almost across the board with all these diseases. Off-label use for providing some clinical benefit. I've talked about the number, the spread of the histological type. Now, what is becoming increasingly interesting to us is beyond histology, what are the genomic underpinnings of this disease? Now we are beginning to learn that actually you could put this disease in different categories based on mutations or some genomic underpinnings. Could you go on to the next slide? And this is where we think LP-184 has an opportunity. So in terms of the challenges, though, the disease is not one. It's very heterogeneous. And I've just talked about these different buckets of the disease. And that is also applicable as we look at the genes that are recurrently mutated. It turns out that as we do a deeper dive into this, clearly LP-184 has an opportunity here because the P10 mutation is one of those aberrations in the DNA damage repair pathways that lends itself to sensitivity to LP-184. And of course, we have preclinical data in rhabdomyosarcoma, various subtypes showing activity of this drug. And again, this is one reason why our current conversation with the Children's Oncology Group is to design a study. In fact, and this week, we are meeting with them in New Orleans. So we have a draft protocol that we've designed, and we want to go with them so that we can obviously start thinking of ways of implementing a personalized medicine approach in this patient population. Now, let me switch gear to hepatoblastoma. This is an embryonal tumor. Again, it's a disease that occurs in the first 2 years of life. It's really the most common liver tumor, malignant liver tumor cancer in pediatric patients. Unfortunately, it happens so young in life. It's not common in the older age group. Could you go on to the next slide? Numbers here are still small, about 50 to 125 cases diagnosed in the US. Now, this is a little bit better in terms of the overall survival, upwards of about, 5-year overall survival, upward of close to about 70%, which is good news. The question we always ask is there room for improvement? And the answer obviously is yes, for the other 30% of patients. So this is where, again, LP-184, comes in. The modality of treatment is similar to what I presented for both Rhabdomyosarcoma and MRTs as well. It's a combination of surgery, radiation, as well as chemotherapy. Now, the interesting thing about this particular disease is that it's extremely, exquisitely sensitive to a drug called cisplatin. So cisplatin is the backbone in combination with 5-FeO4, 5-fluorouracil, as well as vincristine. We are also, like in other indications, beginning to understand beyond just histology what else might be driving this disease. What are the mutations that are very common in a disease such as this? So I'll speak to those in the next slide. Now, in terms of the challenge, I talked about cisplatin being almost the backbone. The disease is very exquisitely sensitive to cisplatin. But here's a problem. Cisplatin causes damage to your hearing. So you picture a child at about 1 year of age or less being treated with a drug for which you are literally thinking, would this patient go deaf the rest of their life? Remember, this is a 1-year-old kid. They have to grow. Obviously, hearing is a critical component. So a disability in that regard, we all believe will have some impact on the growth and development of such a child. So there have been opportunities now and studies trying to look at replacing cisplatin with carboplatin, which is still a platinum agent in that group, but doesn't cause problems with hearing in the patient. Unfortunately, those trials really have not addressed the question of clinical benefit. So increasingly, there's this personalized oncology evaluation to figure out other therapies other than these traditional treatments. In this regard, we know a signaling pathway involved in DNA repair. Mutations in that pathway also are common in this disease. And this is where we think there's some vulnerability as we look at LP-184 in this disease. And indeed, we have preclinical data also that points in this direction. We have some exciting activity and so on. So let me just summarize in saying that these are 3 indications that for which we know the numbers are small. But interestingly, I want to emphasize this across the board. These tend to happen in kids that are much, much younger. Now, you might ask, why is that significant? And what's the relevance to LP-184? It's what we call metabolism of drugs. Most of these drugs I talked about that patients are being treated with, these are little kids. They don't behave like adults. So the way they handle these drugs clearly will be different from adults. And some of these drugs also have what we call drug-drug interaction potentials with other medications that you might want to use in the context of treating a cancer patient. Now, none of these liabilities, as far as we know till date, applies to LP-184. So that makes for a relatively easy development, or at least incorporation of LP-184 in the treatment of these diseases. The other aspect as well is LP-184 is administered intravenously. Yes, the other drugs are treated also intravenously, but once in a while, this is a challenge in drugs that are being developed in pediatrics. You want to make sure that there are no issues with formulation for a kid that is much, much younger. And here we have LP-184, obviously, that is not exposed to that liability as well. So I hope I've given you a good overview. the current challenges in these three indications and why we strongly believe that LP-184 actually has a significant, potentially a significant role in the management of these indications. So let me hand over to Panna now, and maybe at the end, if there are any questions, I will take those.
Thank you. So I'm going to get into the preclinical results, and they're very exciting. Now, some things to remember as we go through the preclinical results is that these data were generated as part of the Pediatric Preclinical Testing Program, the PPTP, and it's sponsored by the National Cancer Institute. And the point of this preclinical testing program is to develop agents that may have activity, significant activity in pediatric cancers. So there are two things that we're going to be looking at. One of those, go to this camera. No, okay. The two things that we're going to continuously talk about in all the models, and this is actual data, very exciting data. The first thing is what is event-free survival. So what does event-free survival mean in this context? Event-free survival means how long does it take for the tumors, which are typically about 200 to 300 or so cubic millimeters at the time, to grow fourfold. That means 4x. And so we, first of all, want to look at how long does it take in the natural setting for these tumors to grow fourfold. And as you can see in the MRT models, this varied from about 14 to 34 days. So that's without any treatment. So what happens when we introduce LP-184? You can see that not only did we have complete tumor regression, and that's very important. It's not like the tumor decreased by 30% or 40% or 50%. We're talking nearly 100%. Now, these mice are, they're given, obviously, these patient-derived malignant rhabdoid tumor cells, and they grow in these mice, and these mice eventually, these tumors get out of control. But in the mice, which we give them 184, they get complete regression, and we measure them, and the tumors never recurred for about over 98 days. And that's the entire duration of the study. So that is pretty extraordinary that in all situations in MRT, which is a very rare and aggressive cancer, as Reggie pointed out, we had very good response, and the response was for the entire duration of the study. Let's look at what we saw in ATRT. And again, this was the first pediatric tumor that we got a rare disease designation. And this is a CNS tumor. And we saw on average about 82 to 90% tumor growth inhibition. Again, very significant. As you can see in the chart, the tumors were completely destroyed, both at 2 mg per kg and at 4 mg per kg. But what you also saw, again, is that there was very good durable response. And we were able to actually even improve the response by introducing another agent called spironolactone. So this is very synergistic. So this is what got us very excited, in particular, about ATRT in the brain, because not only did it cross the blood-brain barrier, not only does it give very significant 80 to 90% decrease in the tumor size, but also that we can actually use a whole nother new agent that's already FDA approved to actually improve the response. And that response is pretty significant. If you look at the charts, we're looking at 30 to 60, 70% improvement in what we think is overall survival as a result of introducing this agent. Let's look at rhabdomyosarcoma. Again, there are multiple types of rhabdos. We had 5 very different models. And again, the event-free survival was 98 plus days. So again, the entire duration of the study. So once the tumors were destroyed, again, and not by 30, 40, 50%, we maintained complete response. So, unfortunately, these weren't actual children, they were mice, but these were patient-derived rhabdomyosarcomas. If you look at the green and red and blue lines, you see these tumors, we had really great response across the board. And more importantly, again, we maintained complete response all the way through the end of the study. And again, same thing in hepatoblastoma. In the controlled mice, the tumor volumes quadrupled in a matter of less than a month, 21 and 28 days. In our cases with LP-184, we saw, again, after 4 doses, we saw complete destruction of the tumor. And again, that was maintained for the course of the study, which is, not a little over 98 days, I guess. So these are really extraordinary data. I mean, so this isn't just in one model. This is across 3 very different cancers, across multiple models with patient-derived material. And we saw complete tumor regression in all of these models and maintained complete response. So this is what gets us excited. This is a wonderful drug now to try. As Reggie pointed out, we think we'll have probably fewer side effects because of the more targeted nature of the drug, and also because the drug seems to be tumor site activated, meaning it really doesn't come alive until it actually gets inside the cancer cell. So we think that also will provide some benefit to minimizing any off-target or side effects as well. So Reggie, thank you for providing us an overview of the disease, and hopefully I gave you some undercurrents about some of the exciting data that got us going. And at this time, we'd like to open up for any questions people might have. So let me read out the first question is, what is the mechanism of action of LP-184 in these pediatric tumors? I'll talk a little bit about what is driving it, and Reggie, feel free to chime in. The mechanism of action, we believe, is it causes double-stranded breaks in the DNA of these cancer cells. And in particular, cancer cells that have shown some damage in the DNA repair pathway. And there are a number of genes and pathways involved, whether it be nucleotide excision repair or homologous repair, or even other DNA damage repair genes like P10 was mentioned earlier. There's another very important hallmark is that as these tumors become more aggressive, there seems to be an upregulation of PTGR1, both as RNA, but also as an enzyme. And when PTGR1 is overexpressed over a certain threshold, we find that 184 is more active. So we believe that PTGR1 overexpression inside the cancer cell, coupled with some kind of DNA damage repair pathway, mutation or deficiency, like SMARCB1, for example, in ATRT and MRT, drives the activity. Reggie, anything to add to that?
No, that's spot on. Nothing additional from my side.
Great. Another question is,
when do you plan to initiate a trial of LP-184 in these indications? I'll take that. Actually, like I said during my presentation, we already have a protocol in place that we're discussing with the Children's Oncology Group. And barring any obstacles, I think by the end of this year or first quarter of next year, we should be able to initiate a pediatric program.
Great. We have time for maybe one more question. Do you plan on combining LP-184 with other therapeutic agents? The quick answer to that is yes. We found a number of remarkably synergistic agents out there. We talked about one of them today, spironolactone, which we think will be very useful in both pediatric as well as in CNS cancers with 184. But 184 has shown synergy with IO agents. It's shown synergy with PARP inhibitors, and actually even with other DNA damaging agents. So there's a number of patents that we've also applied for where we have shown synergy of LP-184 when used with other therapeutic agents. Reggie, anything to add to that?
No, maybe I take just very quickly. I think one of the emerging strategies with the pediatric community, obviously, is to look for ways of taking very early drugs like LP-184, for example, and combine it with standard of care regimen, maybe in an attempt to see if they can have a randomized trial. So that's a strategy also that we are keen on discussing with the Children's Oncology Group as we move forward.
Great. Well, I want to thank everybody for joining today's webinar, especially Umesh and Celine and Rick and Peter, Liza, John, Julia, our team back in Plano. So thank you all. This webinar will be available on the YouTube channel as well. So for people who were not able to join live, it'll be recorded and available on YouTube. Thank you very much, everybody.
All right, thanks.
Thank you, Reggie.